Very good. Thank you very much. And thank you all for coming. I'm very impressed on a nice Friday afternoon. You're all packed in here. I've got good news. There's no test, so don't bother taking notes. And the whole purpose of this is to give you a very high-level overview of a variety of different modalities in cancer therapy. And I have been given the task of talking about immunotherapy today. That's what I do. I am a translational clinical researcher, and I'm going to show you some of the work going on in the immunotherapy arena, hopefully tell you why it's going on and convince you that actually it's the only way we should be treating cancer. And the rest of the conversation today will be irrelevant to you when I'm done. So with that, see, the first rule at the med school, we don't have an AV person. So we have to know how to do this, and we don't have all this fancy equipment, so it usually behaves much nicer. So we're going to talk about a variety of immunotherapy options for cancer therapy. I'm going to sort of give you the history of immunotherapy, and then I'm going to tie in what we're doing today as to why it's an improvement over some of the older immunotherapy treatments. Talk about some novel ways to approach it with combinations of agents. And then we're going to talk about some new agents that have very recently been developed, the rationale for using those, some other agents in development, and some novel approaches. So this is an early, relatively simplistic, although it's 2003, a relatively simplistic view of the immune system. There's actually two different kinds or two different arms of the immune system. You've got your adaptive immune system, which is the one I am interested in. And that really is made up of your lymphocytes for all intents and purposes. You've got B lymphocytes and you have T lymphocytes. B lymphocytes make your antibodies, and T lymphocytes are the ones that are relevant for cellular surveillance as well as treating certain infections like fungi or tuberculosis. In particular, since the T cells are relevant for screening and fighting cancers, there's been a great focus on developing ways to stimulate the T cell arm of the immune system. And interleukin-2, IL-2, is a cytokine. Cytokines drive the proliferation and stimulation of lymphocytes. And interleukin-2 has been around now for really 25 to 30 years. And has been the backbone of immunotherapy for most of my career. And this sort of illustrates a simplistic diagram of what IL-2 does. IL-2 stimulates both NK cells and T cells to proliferate, and when you give it at high dosage, it has a very stimulatory effect. It results in the release of a number of other cytokines. Some of the interferons are released, and you get proliferation of T cells, enhancement of cytotoxicity. There's got to be a better way to do this. And you end up theoretically targeting tumor cells. And the reason it's theoretical is because we dump this into patients. Patients have the toxicity associated with interleukin-2, which is primarily life-threatening hypotension. But exactly why the tumors regress in some patients is unclear. Now, if you look at mouse models, the reason I do immunotherapy is shown in the top slide here. This is a mouse model of breast cancer, actually. Doxorubicin is a chemotherapeutic agent that treats breast cancer. And you'll notice that the control patients, or the control mice with tumor implants do very poorly. Only when you give doxorubicin plus an immunotherapy agent do you get a flattening of the curve and actual cures of mice. Down in the bottom slide, you can see that with any of the single agents, you don't get cure. You really need a combination with immunotherapy to generate cures. Now, this is actually in humans. This is a result of giving single-agent high-dose interleukin-2 to patients. This is actually renal cell patients. And you'll notice two things. First of all, this is a typical survival curve, demonstrates the survival of a cohort of patients. These are only patients who achieved responses. And in cancer, we define a response as more than a 50% shrinkage of disease. That's a partial response. A complete response is total elimination of cancer. Those are very rare in metastatic cancer patients. As a matter of fact, immunotherapy is probably the one model where we cure advanced metastatic disease. These curves illustrate that, for example, the blue dots are patients who achieved a complete remission early on with their therapy, and you'll notice that you won't see many of my colleagues showing you survival curves out to 12 years. There are patients about, where did our AV guy go? About 10 to 20% or so of patients will have very long-term durable remissions, and actually about half the patients who achieve a complete remission will have stable long-term cures, and I've got a number of patients who've been cured of their cancers. It keeps, I don't even touch it, it just, yeah, I'm not doing anything to it. I'm doing the same thing I do every day and never does this. Ah. We'll see if that works. That's a good idea. So with that background, indeed, only about 10 to 20% of patients were long-term survivors, so obviously there's great room for improvement. This was a trial that was recently done looking at high-dose interleukin-2 alone versus combining it with a vaccine. So vaccines, obviously, you're all familiar with. You get flu shots, hopefully, which stimulate your body to be protective against the flu virus. In cancer, we've spent 25 years giving a variety of different cancer vaccines to patients with the hope that we would generate similar immune responses and would actually treat cancer. We have not been very sophisticated in that approach. When I started in the lab, we actually collected colon cancer tumors from patients. We chopped them up in the lab and radiated them, and then we gave them right back to the same patient. And the theory was that perhaps they didn't have an immune response to their tumor when it was in their colon, but by chewing up those tumor cells and injecting it, we would get a better response. Needless to say, that didn't do much for patients, and part of that was because we didn't understand the nuances of the immune system. One of the nuances, of course, is that protein epitopes are handled in the context of your major histocompatibility complexes. You have to have the right MHC complex to handle your antigens. So in this trial, we actually knew that. So the GP100 vaccine is a melanoma epitope, highly conserved on most melanoma patients' tumors. And thus, if you give it to HLA-A2 positive patients, you should engender an immune response. So the theory here was you combine it with IL-2, perhaps we would get better results. There was robust preliminary data, a number of small trials showing that the anti-tumor response rates were much higher when you combined the vaccine with the IL-2, for example, 25% versus 12%, and therefore this trial this approach was taken to a randomized phase 3 clinical trial, 2 arms of patients compared head-to-head prospectively, and the primary endpoint was the response rate or tumor shrinkage rate in patients. A number of secondary approaches were also taken. Patients in this trial, there were approximately 100 patients per arm in this trial. Mean age was about 50 because the major toxicity, again, is life-threatening hypotension, so the patients have to be healthy. Most of the patients were Caucasians and good performance status. In terms of response rate, interleukin-2 alone gives, as I showed you, 12% at the surgery branch at the NIH previously. In this trial, the response rate was about 10%. With the combination, it was 22%, so that certainly is encouraging. That was statistically significant. What was even more impressive, however, was the fact that when we looked at the rate of progression in patients, with IL-2, there had never been a trial which demonstrated an improvement in progression-free survival over standard chemotherapy, for example. This was the first time that an IL-2 containing arm was actually able to show a median improvement in time to progression. Now, if you're looking at the numbers, you'd say, well, gee, that's 1.6 months versus 2.9 months, not very impressive. And indeed, that's the case, but it's a small incremental step forward. This is the overall survival curves for patients in this trial. And historically, median survival for patients with metastatic melanoma is on the order of about 8 to 11 months. Here, we got out to about 17 months in this group of patients. So again, nearly a doubling in that overall survival time, suggesting benefit. And as a matter of fact, this was so beneficial that we published this in the New England Journal of Medicine just in the last several months. Now, as I said, we've learned a lot about the immune system since I showed you that very first early schema. And one of the things we've learned is that the immune system, while able to be stimulated, also is critically regulated so that we don't get an overstimulated immune system, because that's what leads to autoimmunity, diseases like Crohn's disease, ulcerative colitis, a variety of other diseases need to be held in check. So as we give things like interleukin-2, one of the things that we do is we stimulate T regulatory cells. T regulatory cells they can be identified in humans in a variety of different ways. One way is they are CD4 positive, CD25 high expressing. Another way is to look at FOXP3 expression. All of those ways you can use flow cytometry in patients to identify these cells. And we know that these cells interact with the immune system in a variety of different ways. They secrete TGFbeta to suppress the immune system. They also have direct cellular interactions with CD8 cells and cytotoxic T cells to suppress the immune response. So as much IL-2 as we give to patients, one of the effects of IL-2 is to stimulate the production and activation of Tregs so that we stimulate it and then we shut our own immune systems down. And theoretically, depleting patients of Tregs would be a valuable way to approach immunotherapy. This can be done. We can actually monitor many of these things on an ongoing basis in patients. So theoretically, you could give interleukin-2 to proliferate your CD8 and CD4 cells. There's a drug called denileukin diftitox, which is a fusion protein. It combines diphtheria toxin with IL-2 sequences, so it targets IL-2 expressing cells, and it actually kills those cells. Theoretically, giving diphtheria toxin is potentially dangerous, and that's why the link to the IL-2 is there. So the rationale for this trial was that we knew from retrospective studies that patients who had a greater rebound in their lymphocyte count after IL-2 did better. So we wanted to sort of challenge that hypothesis by seeing if we could deplete the Tregs, we would get an enhanced response to treatment. So we actually gave standard high-dose interleukin-2. It's given on a week of treatment with a week of rest in between, but we also then combined different doses and schedules of the denileukin diftitox to try to target Tregs. So this is a flow scattergram from the patients that we looked at, and we didn't have FOXP3 staining initially when we started this work, so we actually used as a surrogate CD25 high expression for that same population. And gratifyingly, when we did have FOXP3, we could show that the cells that were high expressors did express FOXP3. So we thought that we were targeting, indeed, Tregs. This slide is from the manuscript that shows the changes in Treg population. So we have the two different groups of patients, and the top panel is before treatment, in the bottom two boxes is after treatment. And you can see that as an aggregate, we had about 9 or 10% of Tregs in patients circulating pre-treatment. We could reduce it by 50% by giving the denileukin diftitox. The different arms, in the arm where we gave it in between the treatment arms, we actually showed about a 2/3 reduction in Tregs. So we proved one point was that we could deplete Tregs in humans. The other thing that was nice was the fact that our hypothesis was that we would enhance rebound lymphocytosis, and indeed we did that. Control patients were not treated with denileukin diftitox. This was from another high-dose IL-2 trial, and then you can see in the group B patients where we gave the denileukin diftitox pre-IL-2, we actually did get a statistically significant rebound lymphocytosis. That was good. What was even better, however, was that we obviously followed patients for responses, and we actually had a 33% response rate in both small groups of patients, very limited sample size, obviously. And yet, compared to the other data I showed you where response rates were 10 or 20%, here we're up to nearly or over 30% in this trial. We had a number of very high-risk patients as well who historically have not responded, patients with sarcomatoid renal cell, for example. So this was very gratifying and sort of opens the door to perhaps other ways of combining novel agents with this kind of approach. Now, Another way the immune system shuts down is there are a number of, let's see if I can find the pointer here. This is a schematic of antigen-presenting cells interacting with a T cell. The sort of fundamental simplistic approach that we had initially was that the protein epitopes were presented to the T cell in the context of an MHC complex through the T cell receptor, and that's what resulted in the ongoing or the beginnings of the proliferative cycle for T cells. We've learned subsequently that there are a number of what are called co-stimulatory molecules. B7 is a critical antigen which is expressed on APCs. And it turns out early on in the T cell stimulation phase, B7 actually binds to CD28, and that provides a dramatic sort of kick to the immune response, and that's what really drives T cell activation and proliferation. However, as that proliferation and activation is ongoing, the T cells actually begin to upregulate a molecule called CTLA4 and another molecule called PD1. Those molecules preferentially then begin to bind to their ligands. CTLA4 actually competes for B7 and actually is a better, tighter binding molecule than CD28 is, and these signals then shut down the immune system again. And that sort of philosophy is called checkpoint regulation, and it was at least hypothesized that you could perhaps prevent that blocking with those checkpoint regulation proteins with other antibodies. And indeed, there are now antibodies, clinical grade antibodies, available for treatment in patients. Ipilimumab is what's called an anti-CTLA4 antibody. So this cartoon illustrates the interaction process between the APC and the T cell, with the T cell receptor and B7 stimulating the T cells. CTLA4 begins to be expressed, outcompetes CD28 for B7, and you shut down the T cells. The theory here was that the administration of this humanized antibody would actually block the binding. It doesn't activate CTLA4, it just prevents B7 from binding. And if you do this, you would get what's called T cell potentiation. You would stop that inhibition. This was a trial that was presented actually a year ago at our cancer meetings. This was a clinical trial, a randomized phase 3 comparison in patients who had failed prior therapies for melanoma, and it looked at the concept of giving single agent ipilimumab with a placebo versus ipilimumab and the GP100 vaccine versus the vaccine and placebo arm. And that was recognized as being a control arm. GP100 had never shown much benefit as a single agent, but at least it was an active theoretical control arm. This was actually critically important because this was the first trial in melanoma which ever showed overall median survival in the history, actually, of melanoma treatments. So this was great news for patients. What wasn't great news for patients was, again, the results were more modest than we certainly would like. You'll notice that, again, just like the IL-2 curves, there is what's called a tail on the curve. These curves don't go to zero. So some patients seem to have very long-lasting benefits. About 20 to 30% of patients getting ipilimumab will have durable, long-lasting remissions. Interestingly, many of these remissions are actually partial. On CT scans, we can still see disease, and yet it stops growing. Whether that's viable disease is unclear. The ipilimumab alone arm, as well as the vaccine ipilimumab combination, both showed statistically significant decreases in the hazard ratio of death in this trial. In addition, the combination improved the response rate. So if you look at the overall response rate, Ipi and placebo was about 11%, 6% for the combination, and only 1% for the vaccine. So improvement in overall response rate as well as survival. Now, these drugs stimulate the immune system, but these drugs also do such a good job of stimulating the immune system that they cause autoimmunity. So there are a number of patients who develop what are called immune-related adverse events. These include skin rashes, but they can also include fatal, explosive autoimmune toxicity in the skin. Patients died with the development of severe life-threatening diarrhea in these early trials. Patients develop profound endocrine collapse. Their pituitaries are ablated by the body's immune system. So when you use these drugs, these are so effective, you need to be very careful. Monitoring needs to be very rigorous so that if you identify that autoimmunity, you intervene quickly so the patient doesn't die. Now, these results, obviously, as I said, were quite limited in their initial presentation. There actually has been data which looks at the sort of the history of melanoma studies in terms of progression-free and overall survival data. And you'll notice that these are where most studies lie. This study, the arms were better, suggesting that this really is an improvement over our historical approaches. As I said, that was an anti-CTLA-4 antibody. We do have anti-PD-1 antibodies in development. This is a phase 1 study that's actually still ongoing, and it's looking at patients with a variety of heavily pretreated refractory solid tumors and giving this PD-1 blocking antibody. Interestingly, this is very well tolerated, has much less in the way of autoimmunity, it seems, and despite heavily pretreated patients, response rates of 30 to nearly about 30 to 40% have been seen in patients with kidney cancer and melanoma. And interestingly, if you respond despite having failed multiple prior therapies, none of these responders actually have then progressed. So this suggests that this agent is even more potent, and we've just launched a clinical trial looking at different doses of this agent in patients with refractory renal cell carcinoma. So hopefully this will show similar benefits, but more robustly and safer than we saw with the CTLA-4. Now, I'm going to shift gears for a second. Let's talk about true vaccine therapy. So I've alluded to vaccination, and obviously the model for vaccination is an antigen is presented to the immune system. Basically, antigen-presenting cells scavenge some of those antigens, and they become antigen-loaded. They process that antigen, and they convert themselves into dendritic cells, which migrate to your lymph nodes, where they interact with T cells to drive the stimulation process. This is an artificial vaccination process. This was developed for prostate cancer. In this process, instead of getting a vaccine in your arm, the patients actually undergo a procedure where their blood cells are harvested, and some of their mononuclear cells are shipped to a laboratory in New Jersey. And there they're actually cultured in a vat with the antigen of interest. And then these dendritic cells are collected, and they're shipped back to the patient, and the patient undergoes an infusion, much like a blood transfusion. And the theory was that this would stimulate in vivo the T cell activation, which would then attack the tumor cells. There were a number of small trials demonstrating some interesting but puzzling results. So ultimately, this was taken to a large phase 3 trial. Patients were given treatment. You basically get 3 infusions of this, and you're done. You get this early on in your development of metastatic advanced prostate cancer, and then you can get any treatment the doctors wanted. And the patients were all followed for survival. What was interesting was there was absolutely no impact on tumor progression. Patients' blood tests got worse, patients' tumors got bigger, and yet at 3 years there was an improvement in the median survival time, 25 or 26 months versus about 22 months. This was exactly similar to an earlier, smaller phase study that nobody believed. So this drug was then made available by the FDA. It was approved for the treatment. Another way to look at this is with landmark estimates, looking at survival at 2 years, 3 years, or 4 years. And you'll see in the vaccine arm, here there's nearly a 33% reduction in the risk of death at 3 years if you get this vaccination. Now, that seems great. Vaccines, easy, extends life, nice thing. So there's an overall survival benefit, consistent data across a number of studies, almost no side effects to this therapy. Patients, every prostate cancer patient would love to get it. However, this is frustrating to patients because when you measure their PSA, which is a blood test which monitors disease activity, it's still going up. So the patients really don't know if they are benefiting. Certainly true populations benefit, but an individual patient doesn't. The other frustrating part of this is it costs about $100,000. for that 6 weeks of treatment. So it's a little bit of a pricey sort of approach to therapy. Now, I'm going to close with some novel work that's been going on downtown. I mentioned TGF-beta earlier in the context of T regulatory cells, but TGF-beta is secreted also by tumor cells, and it turns out that TGF-beta not only transforming fibroblasts from benign to malignant, but actually turns out to be a potent immunosuppressive agent. And for a long time, immunotherapy people have wanted to look at TGF-beta depletion. This is some data from prostate cancer patients just looking at the risk of recurrence after surgery based on quartiles of TGF-beta in the blood, and you'll notice that patients who have very low levels of TGF-beta do very well. Patients with high levels do much less well and fail very quickly. And therefore, some work that I'm doing with my colleagues downtown, Dr. Chung Lee in urology, is asking the question, if we can render tumor or the immune cells, host immune cells, insensitive to TGF-beta, would we be able to actually eradicate tumors? And we've done a lot of the preclinical work that's needed for this kind of work. There's been some mouse work done. We harvest donor lymphocytes from the spleen of a mouse, and we give it to one of its litter mates, but we give it after we have transfected it with a dominant negative TGF-beta receptor, so it's the CD8 cells are unable to respond to TGF-beta. We actually have a construct which basically has replaced the standard TGF-beta type 2 receptor with this dominant negative, which is lacking the transmembrane and the kinase activation portion, so that the TGF-beta binds, but it doesn't suppress the immune cells. And the theory was this: we would take these donor lymphocytes, we would then grow them or co-culture them with irradiated prostate cancer cells. We would transfect them after we expanded the population of CD8 cells, and then we basically inject them into tumor-bearing, same tumor obviously, mice, and we follow the patients, or the mice in this case. Mice do very well with immunotherapy. We cure lots of mice, and this is an example of that. If you give the wild type CD8 cells and you sacrifice the mice to see what the efficacy was, almost all of these mice have lung metastases at the time of sacrifice. If you give just a sham construct transfection, about half of the mice had lung metastases. If you give the dominant negative CD8 cells, almost none of the mice develop lung metastases. And indeed, you can actually image these with different markers, and you can notice that if you do stains for CD8, with the naive, untreated CD8 cells, almost none infiltrate into the tumor, whereas if you have the dominant negative, the CD8 cells actually penetrate into the tumors. They induce apoptosis, and so we clearly think that the induction of this resistant CD8 cell can induce and generate the results we want. One of the criticisms of cellular therapy for cancer is that in most models where it's been given to patients, the CD8 cells that are transfused fall off very quickly with time, so you don't get long-lasting immunity. What's interesting about the data with the TGF-beta dominant negative is we could detect these cells circulating for very long periods in mice, suggesting that there is a longer, more durable benefit. So I'm going to close with sort of a theory of anti-tumor immune therapy. This is the normal state that we have patients in currently. Patients have tumor antigens that are always available for the immune system to detect and handle with antigen-presenting cells. But the CD4 and the CD8 cells, although they begin to be stimulated, there's a number of different processes active in patients which suppress that immune response, ultimately resulting in tumor progression. And I would like to actually believe that using a different approach where we begin to actually expose the system to cytokines, a variety of cytokines, try to deplete immunosuppressive cellular components, give some of these checkpoint regulation, manipulate the CD8 cells, we can sort of create a cocktail where actually we can kill tumors instead of letting them live and grow, and hopefully will generate some of those long-lasting survivals that we like to see at greater rates than 5 or 10% in these diseases. So just to leave you, multiple new approaches ongoing. Certainly the molecular biology and the tools that we have dramatically changed the way we can understand the system and approach it. Many new agents and novel mechanisms. I've just given you a small sampling of some of those are available. And hopefully, using some of the tools that you guys have available to you, we can perhaps partner and create some novel translational approaches that we could take forward in the future. Thank you very much. Dr. Licht has arrived. John, good to see you.